my name's Amanda Mitchell. I'm a scientist here at the Allen Institute. I've been at the Allen Institute for a little under a year. I started in July of last year. My background is in neuroscience and epigenetics. I've been studying and working in the field for years, really since undergrad. I most recently came from Merck, where I was in early discovery research, doing research there. And I'm originally from the South. I'm from Nashville, Tennessee.
So I'm going to go ahead and share your slides. And when you're ready to move on to the next slide, just say next and I'll advance.
Sure. Great. Thank you. Thank you for doing that for me. So I wanted to talk a little bit about my background and some of the research I've done in the past. And I think the biggest thing for my research is really trying to understand neurotherapeutics. So what are some things that we need to understand so we can get to the point where we can make new treatments for neurological and neuropsychiatric disorders? Next slide. Okay, so this is a little bit of my background. My background is neuroscience and epigenetics and genetics. My PhD is from Vanderbilt in neuroscience. And after that, I did a postdoc at Mount Sinai in neuroepigenetics. While I was there in New York City, I also was an adjunct assistant professor at Yeshiva. I taught neurobiology and psychobiology and behavioral endocrinology. After that, I went to Merck and I worked in early discovery research for neuroscience. And now I'm at the Allen Institute. In my spare time, I love photography. I love traveling. Haven't been able to do that a lot lately, but it's one of my loves. So what I wanted to do was kind of just take you through some of the research I've done along the way from undergrad all the way up to now. So I'm going to talk about one project from each of these different realms, and hopefully this will give you some idea of what I've done and kind of what led me to where I am now. Next slide. Okay, so undergrad. So I went to Vanderbilt for undergrad, and one of the first biology courses I took, one of the professors suggested for all of the undergrads to get involved in undergraduate research. So I decided my sophomore and junior years to do research in the Circadian Rhythm Lab, and I was fortunate enough to have part of my research published. So we were studying the development of the circadian clock, and I was working with a postdoc who was also a pediatrician, and he really wanted to understand how it developed so that when he worked on prenatal infants in his practice, that he would know how to treat them better. So we dissected out the region of the suprachiasmatic nucleus, which is the circadian clock, and we imaged it. So we put it in a dish and imaged it in a microscope. The mice that we were using had a reporter on them. So the expression of one of the clock genes, period 1, was coupled to a GFP reporter. So we could measure individual cells. So like the way the clock is orchestrated is that you have transcription of specific clock genes that oscillate in time. So basically your circadian rhythm is being driven by the oscillation of transcription and of cells. So by measuring this, we could see how the clock was performing in animals. And so we took little mouse pups and we put them in constant light conditions. So that mimics what you see or what was in 2006, what was being used in ICUs. So what we found was really interesting. All of the cells in the SCN under constant light conditions are desynchronized, and you can see the rhythm in the GFP and you can visually see it as well. So this also results in altered behavior. So those mice, they were active all the time. They didn't sleep very well. They slept for a few hours. And so he used this study in his practice so that infants are now given cyclic lighting patterns when they're in the NICU. And part of the reason they were in constant light really was just because doctors are working on them, but it's most beneficial for their behavior if they're in cyclic lighting periods. So from that, I really enjoyed that research, and I did other research during the summer, but it's hard to have a research project be published in the summer, but some of the summer research projects were with different diseases. And what I really came to understand is that you can use research to try to look for new treatments. So up until this point, the way I saw science was, you get your science degree, then you go to med school and you become a doctor. I didn't know that there were like these other options of things that you could do. So from there, I went to graduate school. And so in graduate school, I was in a different lab at Vanderbilt. I took a year off between the two, undergrad and graduate school, because I didn't really know exactly what I wanted to do. So I ended up in a psychiatry lab that was studying a bunch of different diseases. So this lab had postmortem brain tissue from different places across the country. And he had built up this brain bank to study diseases. And he basically wanted to understand gene expression patterns in the diseased brain. So the model that I was able to look at for this is a Parkinson's disease model. So I wanted to give you a little bit of background into Parkinson's disease. So Parkinson's disease is caused by the death of dopaminergic neurons in the substantia nigra. And so this is part of the motor pathway. So when these neurons die, there's a motor phenotype and it's progressive. So there are a few studies at the time that showed that people who exercised had a delayed onset of Parkinson's disease. And even when they got Parkinson's disease, they would do a lot better if they exercised. So we wanted to understand what's causing that molecularly in the brain. Next slide. So we took a rhesus monkey. We looked in rhesus monkeys, and this was done in collaboration with another professor at a different institution, where she had a Parkinson's model in rhesus monkeys. So they're given a chemical called MPTP, which causes the dopaminergic neurons to die. And in this model, these monkeys were just given a unilateral lesion, and then they were exercised. And so we had several different groups of monkeys. Some were exercised, some were sedentary. They all had accelerometers on them, so we could measure their activity patterns. And our final result, we took at the end of the study, we looked at gene expression in different regions of the brain. And we also looked at activity with PET imaging. So the image, the pretty image you see on the bottom is a PET image. And what we found, and this is for the dopamine transporter, what we found is that the monkeys that were highly active, even if they didn't exercise per se, they were just active in their pens, they were protected from the lesion from MPTP. And the signature that I found that was associated with this was in chromatin remodeling genes, neuronal surviving genes, and genes involved in synaptic transmission, which was interesting because we just thought it would be like growth factors, right? That you exercise more, you increase growth factors and increases the survival of neurons. But I was just surprised that it was all these epigenetic factors. And I didn't really understand epigenetics. I understood gene expression. You have genes that are transcribed from DNA, but I didn't really understand what the mechanisms of gene transcription. So that led me to my postdoc. Next slide. And my postdoc was really just studying the neuronal epigenome in disease, neurological and neuropsychiatric disease. And this is basically the mechanism for gene expression. So your DNA has a specific structure in the nucleus. So if you take a chromosome and you unravel it, what you notice is that the DNA is wound around in octamer proteins called histones. And the histone proteins have modifications on their tails. The DNA has modifications. And all of those modifications together can determine whether or not genes are repressed or if they're expressed. And what we found is that the architecture of all of that and the three-dimensional structure changes over the course of development and with experience in both the normal brain and the diseased brain. So I use this information to build a project to understand cognition in the brain. Next slide. And the disease that I've studied in my postdoc was cognition and schizophrenia. And I used a variety of different data sets and I put them together to try to come up with a high-ranking predictor of schizophrenia. I used some genetic data sets, expression, and epigenetic data sets using ChIP-seq. And the gist of what I found is that we found this one regulator that was dysregulated in all three of those data sets called NEF2C. MEF2C is a transcription factor. And I took that gene and I then overexpressed it in the mouse brain. And I found that if you overexpress it, you have improved working memory. So this was a really fun study. It was, taking data that we had, understanding the gene expression, genetics, epigenetics, and then trying to predict, what's downstream of that, what could we do to treat, what genes could we target to treat cognition deficits in schizophrenia. So this led me to where I went next, which is at Merck. Next slide. And that's basically what we were doing at Merck. That's what I did at Merck for 3 years. We were looking at data sets, genetic data sets, to try to identify new gene targets for treatment. This is very, very early stage drug discovery. We were very opportunistic. We were looking at all diseases. We were trying to come up with genes that we could target. And we were also trying to make sure that the genes that we targeted wouldn't have adverse effects for other diseases. Part of the premise of this is it's really, really hard to target the brain. And many of the things that you target aren't effective. There are a few studies that show that if you target something that had human genetic evidence, so basically it's like if you find a gene where there's a familial variant, if you target that variant, you're more likely to treat the disease than anything else. But then again, we don't completely understand the biology of the brain, and we don't really understand these disorders. So I was there for 3 years. There were a number of failures. I was in early stage, so lots of what we were doing were just, they weren't really going to make it to be a drug. Part of that was like, many of these genes are expressed all over the body. So it might be an ion channel in the brain, but it's an ion channel in the heart. So if you inhibit it, you inhibit cardiac function. And so then that's just, you can't go after that target. So it was surprising that there's a number of things that we just couldn't target. And our filtering was really, really stringent. And most of the projects I worked on, I knew would never be drugs. And we would have discussions about, well, what do we do next? How do we make sure that we can really target the brain? And so we had like some ideas of things we needed to do. Part of that was intersect a bunch of different types of data sets, so genetics, epigenetics, gene expression, cell types, to try to really specifically get at different circuits that were dysfunctional in the brain. And it was around this time when we were having some of these discussions with different people that I heard a talk by Hong Hui, who's the executive director of the Allen Institute. And she gave this really beautiful talk about cell types in the brain and the mission of the Allen Institute and the 15-year plan for the Allen Institute for cell types, and how all the teams kind of went together and, they were looking at gene expression and electrophysiology data, and they were putting all of this information together to really understand cell types in the brain. And it was, it was pretty much a lot of the things that we needed to do to really get drugs through the pipeline. But a lot of that research can't be done in pharma. So a lot of the time pharma will partner with startups, institutions, labs to try to get the information that they need. So when they find something that's working in a lab, they might license it from that lab. And so I felt like I was in the wrong place to do what I wanted to do. So there was an opening at the Allen Institute in one of the groups. So I applied and I was offered the position, and I left Merck and came to the Allen Institute. So now what I work on is the classification and characterization of cell types. I work looking at the transcriptome and the epigenome, and I work trying to make genetic tools to specifically target specific cell types and circuits in the brain. I also do a little bit of spatial transcriptomics, where I'm looking at, expression in different layers of the brain. On the right is showing you some clustering data from the nucleus of humans. This is RNA-seq data, single cell and single cell attack-seq data integrated together for cell types. So two of the really most important cell types for that region are the two dopamine receptor, dopamine 1 and 2 receptors. And the two bottom graphs are peak data, ATAC-seq. So this is open chromatin peak data, where there are enhancers. And so what you can do is you can take these open regions of chromatin, you take the DNA sequence, you put it into a virus, and then you can inject that into the mouse, and you can drive expression in that cell type. And that's where I am right now. It's been really exciting so far. We're setting out to do a lot of different experiments and different projects that I think will be beneficial just in general for understanding the brain. And I work in part of a larger team to do all of this. So next slide. I feel like I've done a lot of different things, as you can see, and they probably seem like really different. But when I started, I think my career goal, some people that go into science and are like, I want to be a professor, I want to have my own lab, I want to do this. When I went in, I really just wanted to work on developing novel therapeutics for the brain. And so in my mind, there's so many different ways of doing this. So I've like pretty much done a lot of stuff. Started out in undergrad, just studying a region of the brain. In grad school and in my postdoc, I studied different diseases and linking data with that disease. I worked at Merck, and now I'm at the Allen Institute, which is kind of like it's not academia, it's not biotech, it's kind of like a hybrid of the two. So for me, going forward, I could either stay at the Allen Institute and study cell types. I could join a biotech and develop new ideas. I could go back to pharma and develop novel therapeutics there, or I could go back into academia and study mechanisms. I think any of those for me would be a win. I think ultimately it would be really cool to develop a novel therapeutic for the brain. I think that many people don't have the opportunity to do that, though. I just hope that eventually I can. Last slide. So all the research I've done has been funded by a number of different places, the NIH, and here at the Allen Institute, we're fortunate enough that we have money from Paul Allen to really tackle some of these really big questions. So I hope that provides a background for some of the things I've done, some of my research, and some of my goals and where I hope to go.
Thank you so much for the overview and introduction of your career path. So now what we're going to do is that I have some questions for you, more about your career than about your research. But students, if you want to go ahead and pop your questions into the Q&A button in Zoom, then we will answer those after we go through these starting questions. So, all right, so you've already covered a little bit about what did your science journey look like from high school or college to now, what kinds of positions you've had, and what kinds of research that you've worked on. So can you tell us about what an average day looks like for you, both now at the Institute, which has been entirely in COVID times, and before the Institute in your previous work?
Sure. So before, when I was at Merck, my average day would be, and Merck is a little bit different, I think, but my average day would have been, I would go into lab. I had an office outside of the lab. I'd go into lab, start experiment. So usually I'd have like a project plan for the day or for the week, and I'd start working on experiments. In the mix of that, there would be some meetings. A lot of the meetings at Merck were virtual meetings because a lot of the sites are across the world, and kind of do both. Part of it would be like talking with people to organize the different things we wanted to do. And then part of it would be going in and doing some of the experiments or looking at some of the data. And so that's pretty similar to a lot of what I do right now, actually. I've been pretty hybrid. I do some computational biology, I do some experiments. So what my typical day would be like, an experimental day would be, I would go into the Institute and I'd have some type of project plan that I'd be working on, and I'd set up those experiments and do those experiments. In the mix of that, there would be meetings with other scientists and other people at the Allen Institute around what are our results, what are our goals, where do we want to go from here? And a lot of those meetings are virtual, but I in the past, I was used to having a lot of virtual meetings. For an analysis day, it would be, pretty similar. There would be like a project plan of what I would like to accomplish that day. And then I'd sit down and do the analysis and then get the results and report back to different people in the group and then the team. So a lot of it is having some type of project plan that we're working on, going and doing that research, and then reporting back to other people on what we've done. And that's been at the Allen Institute. That's also been at Merck, a typical day.
So what is your favorite thing about your job? So if we can maybe start with your favorite thing about your work at the Allen Institute and then any standouts from prior positions.
Sure. I think one of my favorite things is looking at the data. We have a lot of cell types data for different regions of the brain. We have a lot of different types of data. And a lot of this data is really new and novel. So it's really interesting to look at some of the cell types, and we've been working on looking at some of the basal ganglia and thalamus cell types. And a lot of that data is just very new. That is probably one of my favorite things. And then I get to do a little bit of mechanism, like trying to figure out mechanisms for different things that are occurring. I also really enjoy that. Just trying to figure out what's happening or what's a theory for what could be happening.
So you mentioned, especially at your time at Merck, that you were in this early discovery period and that a lot of things you worked on were just never going to move into later stages of the development process. And that happens in science, that not everything works the way we anticipate. So can you tell us about your best science failure? Best learning experience, funniest, most surprising thing?
Sure. It's funny now. It wasn't funny then, but I can laugh about it today. So when this was before graduate school. This was the year between undergrad and graduate school. I was working on microarrays in a lab. And, again, some people work with microarrays, but they're old technology now. But this particular lab, he was saving all of his microarrays. And and I didn't know this. So all of the experiments he had run, he had kept the hybridizations of each of them. And I don't know what he was planning on doing with them, because there's really nothing you can do with them now. But we had a sequencing facility that we had a core that would do the hybridizations. And so for one of the experiments, we ran the experiment, but we didn't keep the hybridizations. That was actually a really big failure in the lab. The PI was extremely upset that we didn't keep those precious samples. Extremely upset. So I remember that day I went home and I felt like a science failure. Like I felt like, oh my gosh, maybe I should just quit science altogether. Maybe this isn't it. And so usually if something like that happens or you have like a science failure, what I do is I close my computer, I go home, and I don't think about science for the rest of the day. Like that's typically what I do. That day, I decided to go out with some friends and see a movie, just because I didn't want to spend any more time stressed out about that. The movie that I went to is called Meet the Robinsons. I don't know if you've seen that movie before. It's a cute little animated Pixar movie where this little scientist, at the time I didn't know he was a scientist. I don't want to give the movie away, but basically he's a scientist. He has all these failures that happen in his science experiments, and he might be like 11 or 12. He's not like, he's really, really young. And he's at this point, he's like, I think I'm just gonna quit. And so some things happen, and then everybody tells him, no, you just have to keep moving forward in your failures. You're gonna have failures, but in those failures you learn. And you take the things you learned from the failures and you use them for the next thing that you need to do. So he continues on a science journey, and the little movie, it all ends well for him. So that was, I think, my biggest failure. It seems like a simple failure, but for me, it was like a tragedy. So that's kind of what I do with my failures. I really try to look at what went wrong, but then at the end of the day, I pretty much just close the computer and do something else so that I'm not stressed out about that at home.
We have some questions from students about your range of experiences before you came to the Institute. We have a question. With your diverse background in different fields related to neuroscience before your work at the Allen Institute, what do you believe was the most beneficial experience or something that sticks out to you the most when looking back?
I actually think this is interesting. It's an interesting question because I think I could come up with a few things. I think right now, one of the most beneficial was actually teaching. I taught psychobiology and neurobiology and behavioral endocrinology, and I taught all these things that were so outside of what I did, but kind of gave me a holistic view. And, I took those courses, but, I focused, my research is focused on something else. But I think teaching those courses helped me to understand some of the interconnections between the bigger picture between a lot of things that I've worked on in the past. And I feel like that today has been the most beneficial. Because at the Allen Institute, it's interesting because I think sometimes you think you're going to be in one area, but you end up going into other areas. So having some just background knowledge generally across other brain regions has been really helpful. I think the second thing that's helped me, especially in the environment today, is not having a neuroscience background, but also having a computational background. So a lot of the work that we do is computational, and even at other places, a lot of times people will download data sets from other institutions and try to come up with a new question and analyze it in a particular way. So I actually think both of those things. So having teaching, which gave me a broader background in neuroscience than what I had, and then I think also adding to that a little bit of skill set in computational biology.
I would second that. Programming skills can't be underrated. They're so useful. So another question on the topic of your having many different experiences in your background. You've done a lot throughout your career, and it sounds like they were all extremely beneficial to you reaching your overall goal. Were there any cons to trying out different positions that were not directly related to your goal?
Cons. I'm sure that I'm sure that there's always, I don't know, maybe right out of graduate school, I could have gone straight to something else. I don't know.
There might be none.
I'm trying to think. There must be at least one. I did one summer. I did research in the physics lab. I've never used that ever. I don't think I will ever use it. I think I don't see it as a wasted summer necessarily. I was an undergrad and I didn't really know what I wanted to do, but I think what it helped me identify what I didn't want to do, and there's no need to go back and do that again now that I had that summer experience in that lab. I think I don't know. I just remember being an undergrad. There were things that I enjoyed, but there were also things I didn't know, and I felt like I wanted to understand and explore a lot of different things. So I don't necessarily regret any of those because I think some of them helped me identify what I didn't want to do. Some of them gave me other experience.
What do you think are the most important lab skills needed outside of academia or in academia as well?
Most important lab skills. So right now there's a lot of interest in startups and biotech for neuroscience. And people are really interested in getting their new technology into the brain. So there are lots of places, at least I know of in Boston, that are hiring people who understand models. So understand different models for different diseases. And basically they want people who can model neurological diseases and test their compound or test their new technology. Like, for example, Alnylam, which is using siRNA. I know they're moving right now into neuro. They have no background in neuro. And so they want people who know how to do those models. So I think one of the biggest skills is, at least in biotech and startups, is understanding how to model diseases in animals or in cells. So I would say like understanding how to do, like how would you model Parkinson's disease in cells, or how would you model Parkinson's disease in iPSC neurons, or what type of animal model would you use? That's something that neuroscience is so specialized. I feel like you have to have some of a background or experience to do a lot of it. And I know that a lot of people are looking for that.
So I think the answer to this question is going to vary across the different industries that you've worked in. But how do you know when your research is done? What do you do when you feel like a project is complete? And sometimes the answer is it's never done.
What do you do when a project is complete? So in academia, you would just publish it. So there are lots of grants that end with a publication, and you would publish it, and maybe there's another grant on a different project, and you just move on to that. In pharma and biotech, a lot of project decisions are not made by the scientists, they're made by the company. So somebody will tell you it's done. And it'll be done, and you won't be able to do anything. It'll just be done. There's a lot of, I felt like at Merck, there was a lot of movement of projects from different places. And, based on where the company wanted to go. When I was at Merck, there were a few different pharmaceutical companies that completely got rid of their neuroscience department. Pfizer did, there were several completely. So anybody in neuroscience, not only were the projects cut, but a lot of the people were cut. And so there are a few things that happen, like if a project's cut and somebody tells you it's done in one of those industries, they might put you to work on something else. And so if you have flexible skills, you can move to those other projects. I know some companies, when COVID-19 hit, they moved people to work on COVID-19 projects. So I think in pharma, having flexibility, so having skills in other areas helps a lot when things like that happen.
Yeah, and I would say that's true in academia as well. Like, a paper can sort of mark an endpoint or a milestone in a project, and then you can maybe pivot and bring in some new skills or perspectives. And especially like computational perspectives can be really flexible. So we have another one about sort of research outcomes. Have you had any experiences during your research where you discovered something that you didn't expect? And if so, what have you found?
That's an interesting question. I would say in the postdoc, one of the things that we found that I didn't expect is that a lot of the key genes for targeting neuropsychiatric disorder were transcription factors or epigenetic modulators. And I didn't expect that. Looking back, it seems obvious, but it didn't. It really didn't at the time. I thought most of what we needed to target were signaling molecules, because that's where all the research was. That's where all the research was. And then there was like this shift, and suddenly we started to understand some of these other molecules better. Yeah.
Where did your passion for genetics and neuropsychiatric diseases and your general sort of line of research through these positions come from?
I have always really loved psychology. And so neuroscience is like the biology of psychology. So I really understand, I like to understand why we are the way we are. Why do people do things that they do? And trying to understand those mechanisms for things. That's one of actually the things I do in my spare time is I read psychology books on behavior and economic books on behavior. And I think I've just always been generally interested in that. It's interesting. I think when I first started undergrad, I thought I was going to be a psychologist. The other interesting thing, though, is in high school, so they put all the seniors, they put what they wanted to do in our local paper. And my parents kept that. They kept the clipping of that. And one of the last times when I was at home, I was looking at it and I put down that I wanted to study neuroscience. So I think at some level, I've always just been fascinated with behavior in general and psychology.
So on the topic of reading about research, the field of neuroscience and genetics is constantly growing and more research is published every day. So we have to do a lot of work to keep ourselves up to date on these research findings. And if you can talk both about, what does this look like for you in your current research, and also, how do you choose what to read about for fun, for like psychology or economics or other sort of related things? How does the learning process look like at this point in your career?
Sure. So I think one of the unique things about the Allen Institute is all the collaborations that happen. Just recently, there are lots of different discussions with scientists really across the world about what they think the major questions are for neuroscience for the next 10 years. And I sat in some of those meetings and just listened to them. And so I feel like part of right now, some of the things I'm reading are coming from the leadership and some of the things that they're interested in, and trying to see how they think about problems and questions, and then trying to also to ask them what they think about certain things to get their thoughts on it. From psychology, some of the books I've been interested in, I just look like in the New York Times for bestsellers and things like psychology and neuroscience bestsellers, and then read them. The part of the reason I started doing this is I don't know if you guys have had this experience where you're with some old friends and they're going to law school or whatever it is that they're doing, and they ask you a question about something that you have no idea about, and then they take whatever it is that you said, because you're a scientist, and they take it to heart. And I've had people ask questions about things that like I hadn't really thought about before, but I felt like I need to know the answer to that. Like I need to be able to give people an answer that is logical and that makes sense. And in order to do that, I think I need to understand some of the other things that people are thinking in popular science. Because I think, if you're in like a different field, a lot of people will read some of these like popular nonfiction books. And I was just reading like journals. I was, reading the direct scientific literature. So I started reading some of those popular nonfiction books just so I had a general idea of what non-scientists who are interested in science are reading. And then a lot of the papers like from journals that I read are things that we're working on, things that come out in different journals. Like sometimes I might just sit down and go through Nature Neuroscience and see like what's the latest things that that are coming out in Nature Neuroscience. So yeah, I feel like, and I think the other thing is one of the members of my committee said I should read more. My graduate committee, she said that, and I took that to heart, and I started to read more, and I started to read a lot of different things. But mostly it's like stuff I'm interested in, which helps. It's like if you're in a field that you really like, and it's easy to sometimes pick up some of these books and start reading it and try to understand it.
So what individuals, organizations, or projects have been most influential in fostering your career path and helping you get into where you are today? Perhaps that committee member.
Yeah, that committee member. I think one of the most influential people was growing up, my best friend's mother. So they lived across the street. We were like best friends. Her mom was a psychologist, a PhD level psychologist. And she would always just encourage us to do different things. Like even for like college, she's like, oh, you should look at these schools. You should think about this or think about that. And I feel like she influenced my trajectory in a huge way that I don't think I really fully understand. But I think that was one of the major people. Other people are like professors. So freshman year, one of the first biology professors I had, he really suggested that we get into research. And he was very enthusiastic about everything in research. He really enjoyed what he did. And so he was trying to bring other people into what he did. I still keep in touch with him. It's been like 15 years or something. I emailed him earlier this year about something. And oh, the other thing, I think are my PIs from graduate school and my postdoc, they've been very influential. I feel like, because a lot of my friends have left science at different levels, like after their PhD or after their postdoc. And I don't think I fully understood why they left, but the labs I've been in have been really great labs. And I've had really great mentors who would give me advice and suggestions on different things to do. My graduate advisor pretty much, he advised me. I mean, I asked him, like, where do you think I should do a postdoc? And he gave me a list of people he thought were really helpful and useful and innovative. And so I ended up going to a really good postdoc lab. We were really productive and people were really happy there. And these labs also were like 8 to 5 labs, which is unusual. And so I think my friends who left science were in really bad labs with really bad mentors. So I think the people that you work with make a huge difference in where you go, and their vision, how they work with other people, their vision for where they want to go makes a huge difference. That's also part of the reason why I came to the Allen Institute, was because of the vision. I can see where we're going and it makes sense. And the leadership is very intelligent. So that was part of the reason I really wanted to come here. Yeah.
Yeah, one of the best pieces of advice I was ever given was, if you're an undergrad, a grad student, a postdoc, whatever level, pick a mentor, pick an advisor, not a line of research. You can find an interesting project in many different areas of work, but you need to find people who will support you and be great mentors.
Mm-hmm.
Yeah. And I think that's related to this question. What advice would you give your younger self at the start of your career?
What advice? That's an interesting question. I don't know.
Or to put it a different way that another student has asked, what do you know now about working as a scientist that you wish you had known when you were early in your career as a college student?
Thank you. Yeah, that's helpful. I really like science, right? I love science, and I'm probably like the most introverted scientist. There's a lot of things I don't like, but I think one of the things that I felt like I needed to know that I really didn't spend the time I should have spent on was how to communicate and interact with other people. So at Merck, the teams we were working with so many different people from different areas. So pharmacology, chemistry, I don't know, there may be like 10 different areas. So I was working with a lot of different people. And to really be effective in some of the things that we're doing, you had to be really good at navigating relationships and communicating the information that you have with other people succinctly. So if you can get the information you need to say down to like one slide or 2 sentences, and the other person who's in the completely different field from you can understand it, that is perfect. I think as well, even at the Allen Institute, having projects mapped out. So like, these are our objectives, these are the steps, this is what the result's gonna look like, and being able to communicate that to other people succinctly, because as well, Allen is a lot of teams working together. Been working with a lot of different people on different things, and everybody comes in with a different specialty and expertise. So you have to be able to communicate that to people effectively. I don't think I've been as good at that as I wish I had been. I think in general, in the field, academia, biotech industry, there are a lot of people. I can't say a lot. I can say, well, yeah, I feel like some of the people who do really well, who get up to like really high leadership positions and do really well, are the people who communicate well. They're not necessarily always they communicate and get their information to other people very, very well and navigate relationships very well. Because I felt like that skill, that's like a leadership skill that then helps you lead other people and help people understand what it is that you're doing and help them want to follow you. That's something that isn't taught in science classes. It's not part of the curriculum, but I feel like, I don't know, it should be called something like career skills or something, career communication skills. So right before I left my postdoc, I took a course at the New York, yes, social emotional learning. that is the course I wish I had taken at the very beginning, because it's really important. Yeah, it's just really important. They did have a course like that right before I left my postdoc that I took that helped with some of that. Yeah.
So I have a couple of related questions. If you were to find find an answer in your current project, like, get through the next stage of the project, what would you work on next? Or to put it a different way, if you had unlimited resources, what kind of research question or project would you want to work on? Sort of related questions.
Yeah, if I could do anything, I think I would work on epigenetic projects. So for things like autism, there are a number of genetic genes, genetic associations with autism that are chromatin remodeling factors that we don't completely understand how they work. And part of that is just the complexity of the brain, like, where are they expressed, what are they communicating with? I think I would try to tease apart those complexes, and I would try to find a way to tease them apart. If I had unlimited resources and could do that, that's what I would do. Yeah.
Yeah. So returning to sort of the topic of the many different things you've worked on in your career, has it been difficult to switch between different research topics or techniques, both within a single position, working on, obviously we don't spend our whole time on a single thing in our day, and moving between positions?
Not really, actually. I think I've really enjoyed working on a lot of different things, actually. So doing experiments, doing some computational work as well. I enjoy switching between tasks like that. And even like switching from different places. I've also enjoyed that. So when I was a postdoc, I was also teaching at Yeshiva University. And I enjoyed the time away from being fully in the lab to teach undergrads. I really, really enjoyed that. I think for me, I find it helpful to take a step away from different topics and do something else, because I feel like I process the information better, and then I can come back to it with a fresh mind and fresh perspective on it and do more with it. However, I will say that sometimes people just want you to stick to one thing. Like we just want you to do these 6 things, or, this one thing. But I really do like the switching between things, which I think is why I've been doing that so much.
Yeah, I also find that it helps to just be able to work on something else for a while. And when you come back to that project, you have, refreshed from working on something else and time to process.
Mm-hmm.
So the next question is, have you encountered sexism or other bias in your career? And how have you tackled that? How have you overcome it or found support?
Mm-hmm. So personally, I don't know how much of it I've encountered. I have had friends who've encountered it. I've seen people encounter it. And I think for the graduate students who have encountered it, I think the way that they approached it is they would first go to their PI, or if not their PI, another PI, to try to discuss what was going on. And to try to find solutions to work with it. So I think that was like the first step. And yeah, I think what I've encountered the most from people is sexism, which is interesting. I mean, there are all these different conferences going on, and there are different things going on at those conferences, different weird things going on. that I've had friends encounter. I think the first thing that they've tried to do is to find somebody at the either a PI or somebody at the institution to talk to about what's going on and to find a solution for it. So I was at Vanderbilt, and Vanderbilt, one of the faculty members at Vanderbilt, her name was Beth McLaughlin. I don't know if you guys know her. She started this on Twitter. She started like a Women in Science. She's an interesting person, but really started the conversation about sexism in science. The other thing, so what was the other part of the question? Was it like racism or discrimination in science?
Yeah, and how you have tackled that, found support. Yeah.
So for discrimination in science, it's always in the back of my mind sometimes during meetings or conversations with people. It's always in the back of my mind because the question is, in some of those situations where things happen, you're trying to figure out what is going on. Why is this person behaving in this way towards me, right? And what I've tried to do is to reach out to other colleagues or friends and talk with them about, what is going on here, what could be some solutions to some of these things. And I think one of the best solutions that somebody gave me was he told me, well, first of all, I mean, it's hard to, some of those situations are really hard because it's you in general, and some of the things you're experiencing this from are from people who are in power. And so I don't know that there's a lot you can do. But one of the things I've found that has helped for my career in general is, so every meeting you go to, every presentation you give, if you're in front of like the VP or the executive director or the chair of the department, you're probably not presenting to that person very much, maybe like once a year at most. So the best thing that you can do in those meetings is to be over prepared, to know every answer to every question that they're going to ask you. And to try to always present yourself in that way if you can. And that's your best shot at some of those things. The other thing, so if you find yourself in a place where the environment is bad and you feel like, I don't really have the best solutions to this, but I feel like one of the best things you can do if you find yourself in that type of situation, and it's people in power who are doing some of this stuff, is to find somewhere else to go. There's a lot of places that are very supportive, a lot of different institutions and people that you can work with. And I think without knowing really how to combat some of the things, I think my best advice is to choose who you're working with to the best of your ability and work with people who have, basically, if you can see that they've had other people under them who have been really successful in things that they've done, then typically that environment's a good working environment. I don't really know how to combat some of the issues that exist. I just know that if you can choose good environments and go to those places where people have been successful, you're more likely to succeed. That's not the best answer to that question.
It's hard. There isn't always a good answer. Sometimes you find yourself up against an institutional or systemic problem, and the only solutions that are made available are individual, and that's just not going to fix it.
Right. So some of these places like Mount Sinai Icahn School of Medicine, they have a diversity council, and they now have like quarterly topics. They cover different topics, women in science, diversity in science, and they bring in different people to speak about those things, and they have those meetings together. So some of these places, they have like their trustees about how they're going to address some of these things. I think one of the most important things is to go to a place that has something like that established. It is really hard to try to establish that on your own without support. So before like you go to that school, you can see if they have a board like that. Vanderbilt does as well. Vanderbilt for the longest time has had different seminars on bias, unconscious bias, where they will invite anybody who wants to come, who's part of Vanderbilt, can come to these seminars. And I feel like those have been really helpful. I think the best thing you can do is try to put yourself, if you can, in that environment. It's really hard to try to challenge some of those systems by yourself.
One of the things that you can do to take care of yourself is thinking about your work-life balance. That doesn't solve the institutional problems, but it is really important to do that, as you've mentioned. So our next question is, do you have busy seasons, or is everything pretty evenly distributed through the year? And what do you find helpful in keeping a balance between your work and your personal life?
So yeah, there's some really busy moments. I think what I try to do is, if there's like some type of project that's like important for, really, really important for whatever's going on, I try to focus as much of my time as possible on that project, and then have other moments where I can have more balance. That's probably not the best answer. But then again, I think also some of the places I've worked have really been like 8 to 5, 9 to 5 places, which has helped. I've tried to choose places that have been like that, because I didn't want to be, in lab every Saturday and Sunday working on different things. So I honestly, I feel like one of the best things is try to just choose an environment where people typically have a work-life balance. Like, see what other people are doing, and if they're doing it in that environment, then you'll probably be able to do it. The other thing I think that's helpful for boundaries, work-life balance, is if after a certain time you just don't answer emails. People will stop emailing you after that time because they know that you're not going to answer that email into the morning. Or just like shutting down my computer at a certain time of the day and just done now. I think for me, it's also beneficial to do that because it helps to clear my mind with what I'm thinking about for the project.
And can you talk a little bit about what you like to do for fun, to decompress outside of work?
Sure. So I love photography. I love travel in normal times. So I love photography. So I love taking pictures. I also really love running. I've run a few half marathons, and I've run the New York City Marathon. In addition to that, like with training for some of the marathons and the half marathons, I've done a lot of other exercise in between that too.
So we've talked about how choosing, a mentor or an advisor more as being like one of the most important things that you can do. Have you ever had a time where you found something that was different from what your advisor or mentor was saying? And how did you go about figuring that out?
Yeah. So what this happens a lot though. So a lot of times, so I guess there's two different angles to that. One, there's like the science angle, like your mentor tells you work on this project. This is what I think, and you might find that something different exists. That's one. I think the other thing is like just being moved into different projects or different areas. For the first one, what I've been trying to do as like, and you're more deeply into a lot of the things that you're working on than anybody else, so you know them a lot more clearly if you're given that specific project. What I've tried to do in the past is just to, whatever results I get back, just to try to, as succinctly as possible, diagram out, this is what we found, this is what we did, this is what we found. Here's what I think it could mean. However, what do you think? So trying just to have like the person have the same information that I have, and then to see what they think about it. Because sometimes, maybe there's a part of it that you didn't have that might help the story a little bit more. Or maybe there's something that you saw that they didn't have. So I think just having trying to put together that information so that they can see it. And then for project switches, the second part of that, the different places I've worked at, project switches happen. They happen fairly. They happen a lot. I think one thing that has helped with project switches is just to try to have a discussion with whoever you're working with at the start about objectives and expectations. And then if there's like some type of change or something, you could go back to the objectives and expectations and and say, we started here and now we're here. Do you want to switch to here? Just try to clarify, like, are you, is this what we want to be, is this what you want me to do? And then just go with it. I think that's, sometimes I just feel like the most important thing is just trying to communicate with whoever you're working with about what you're seeing and some of the evidence that you're seeing, and then kind of what does that mean? And then asking them, like, what do they think? Usually I find that's the best way, just to ask whoever, to ask your mentor and your PI, what do you think? This is what I see, but what do you think? Because sometimes there's information that they might have that you're missing.
Oh, missed the mute button there. We do have time for a couple more questions. So students, if you have any more questions, please go ahead and type those in. We did have one quick one about your Parkinson's research. You mentioned that there was a difference related to exercise. What kind of exercise?
That's a good question. At first we thought it was just, so the animals were run on treadmills, and at first we thought it was exercise, 1 hour of treadmill running a day, cardiovascular exercise. But what we actually found is it wasn't that at all. It was just being physically active. It was like, even the studies show that just walking, it's not really vigorous exercise. It's not cardiovascular exercise. It's just being active. It's like getting your daily counts in for the day, really, which is interesting because that could have a genetic component. And so we weren't ever really to tease apart what it was that was causing it, but the animals that were more active were more protective. Whether that was genetic predisposition or by their own choice, I don't know.
Somebody else's future dissertation.
Yes.
So we are almost at time. Do you have any final thoughts, piece of advice, suggestions, thoughts for students' futures as they are looking towards the rest of college and beyond?
Sure. So I think I remember when I was a senior in college, all my friends were like applying to med school and stuff, and I was unsure of what I wanted to do. I mean, I knew there were things I liked, but I didn't know what I wanted to do. And again, I'm a millennial, so this might be a millennial thing as well. I took a year off to try to figure out what I wanted to do. And I think if you know where you want to go, you can get there. But if you don't know where you want to go, and you might go and do thing A because somebody wants you to do thing A, but you might not really like it. So I feel like it's important it's okay, I think, senior year, if you don't really know what you want to do, I think it's okay to spend some time and try to decide. There's a lot of opportunities as well. I think as well, I kind of had this idea that there are only these like 6 things I could do, but there's so many opportunities. A lot of my friends with PhDs are doing so many different things. Some are in finance with a PhD in science. Some are doing consulting. Some have their own labs. Some are working for startups. There's a lot of things that you can do, and you don't have to have it all figured out right now. So.
Nice to meet you all, students, virtually.
Nice to meet you.
Lots of thank you messages. Thank you. We can't see or hear you, but feeling the thank yous.
Thank you for having me. It was nice to be here.
Yeah.